Then I found out about bioprinting, and I thought this is huge. The human impact that you can create and the technical challenge is great. I really love challenges. So I quit my job and create the first prototype of the bioprinter, combining different parts of office printers, conventional printers, a scanner and an old disk reader. So we have that model back in our lab as a museum.
Under the glass.
Yeah, that's right. To remember where we started.
How are you doing bioprinting?
The bioprinter already have the syringe with the patient cells. And then the bioprinter, layer by layer, builds geometry. After that, you have to mature the tissue. So that is going to go to an incubator, and we are going to give this tissue all the conditions that needs. You can use stem cells, or you can convert a cell that is already differentiated to a pluripotent cell, and then reconvert it to another cell type. Usually this is done with fat tissue.
I always thought that fat is a bad thing, but actually it's a very valuable reserve that in the future can be used to give yourself a third ear or lung or whatever, and all of that because you were collecting and accumulating fat over the years.
That's right.
What a relief.
Every year we make a new model of the bioprinter based on the feedback that we get from the users. And what we found out was the academia wasn't going as fast as I would like to. So that is why we set up our tissue engineering lab in Argentina, and we started working on the actual application of the technology. We kept selling the bioprinters, but we started doing research in-house in two main applications. One of them is cartilage and the other is human epidermis.
Who owned the cells that you were using before you got an actual permission to take the cells from a patient? Were they yours?
At first it were my tissue. So once I did a biopsy to myself.
Oh my God.
Because the people that were working at the lab, they were impressed by blood, so I had to do it myself. But we started, the first cells that we got were from me.
Scientists can go to great lengths to make their discovery.
That's right. We are fully committed. Hi, Maria. Thank you. Thank you for having me. Good morning.
Thank you for coming. Yeah, and good evening from stormy Berlin. It's super nice to have you here, and I'm super excited to talk about your passion and area of expertise, that is bioprinting.
That's right.
So when a person who is not a scientist outside from a scientific field, he hears bioprinting, usually they imagine something from the serious West world, full functioning human bodies created with a 3D printer. So what are the real current capabilities that bioprinting has?
All right. So the main goal of bioprinting is actually get to the fabrication of human tissue and organs. Our main objective is for patients in the waiting list, but there are broad applications of this technology. Right now, the technology is in a very early stage. There are some clinical applications, but it's not a gold standard, and that is our main goal as a company, to create the biofabrication or the bioprinting combined with other technologies as the gold standard for patients in the waiting list. So they don't have to wait for a donor, and they don't have to be immunosuppressed, or at least not at the level that they have to be when they get a tissue or an organ from another patient. So the main goal is that in the short term, we are working on more simpler tissues or other applications of human tissue fabricated in the lab.
What can you bioprint right now? Like, I don't know, skin and liver, where we are on this,
Yeah, for sure. Mainly, I think the the concept of bioprinting is actually the process of 3D printing, the conventional 3D printing, where you extrude material layer by layer. There are different technologies, but basically it's layer by layer. So whatever material that you put, you create the structure. It can be plastic, and that is conventional 3D printing. It can be any material, any gel combined with vitamins and different things for printing food, for example. And in our case, we usually use gels, biocompatible gels, charged with the cells of the tissue that we want to create. For example, if you want to create skin, you have different layers on the skin with different cell types. So as you move forward to more complex organs and tissues, the more cell types that you are going to need. Right now, we are focusing on human cartilage for nose and ear. There is the short-term application. It's a very simple tissue. Then we are moving to skin. We are working on them in parallel. But when you go to skin, you have to work not only on putting the right cells where they have to be, but also make the vascularization of this tissue, because this tissue is not going to be only in the lab. It has to be vascularized.
Vascularized is when it's connected to blood vessels.
That's right. Exactly. So the cells, to be alive, they need nutrients. They need the blood. That in the lab, what we use is cell culture media that mimics the blood. And we change the cell culture media every 1 or 2 days, trying to mimic what happens in the human tissue. But when it goes to the actual patient, you have to make sure that it's going to be work in there, and no one's going to be changing the media. It has to be viable.
So it's still in a very early stage. Yeah, we know
right.
how to print some tissues. We still are not ready to print full organs, so can we?
Not yet.
Not yet.
There are some challenges regarding technology in some parts, regarding vascularization. That is the main issue. And also there are ethical implications of new technologies, right? So regulation is not regulated yet. We are very into the human and social impact of our technology. So we do social and training seminars and trying to get the opinion of the people of the application of this technology. And for one of those meetings, we were studying how is the state of the art regarding regulation and the ethical implications of the technology. And one thing that was interesting is who's gonna own the tissue, the intellectual property or the actual property of the tissue that you fabricate in the lab. Because the cells that you are using are from the actual patient, but you are building it like a medical device. And the regulation is a combination between medical device and actual human living tissue. So it's very challenging regarding the ethical implications, the regulation that is not yet. So it's a nice challenge in different parts.
Yes, it sounds really complex. I mean, if you took my cells to make me a new heart, who owns this heart? Technically, this is my heart. But do I need to pay you for a service of printing it, or am I buying it from you? So what actually are the current opinions? I don't know, like which government is the most forward thinking in this regard?
Our experience is in Argentina, so in South America. So in there, talking with the regulatory bodies, they are very, very open to new technologies, and even they've been part of the courses that we give on 3D bioprinting. They want to stay in the cutting edge technology and regulate that. In Argentina in particular, there are two regulatory bodies. It's not like the FDA in the US. We have like an FDA and another regulatory body for living tissue, human living tissue. So there are two different bodies for regulate something that is going to be in the middle. It's very early yet, but it's good to start having those conversations in the early stage. Mostly because we are a company trying to get this to a patient. So we cannot work only on the technology and get to a moment where we have the solution, but there is no regulation or the regulation is going to limit us to get it to a patient.
Obviously, I wonder what was the journey that led to creation of such entity? I mean, an elemental body that is responsible for living tissues. This is something very uncommon. Do you know how it
Yeah,
happened, how they started?
yeah, it's very different from the things that we are learning here in the US. For example, here you have tissue banks where you can buy human tissue for research, only for research. But in Argentina, it's not legal to buy human tissue for any application. You cannot buy human tissue. So what we had to do is to go through a process where a body and a hospital body, a regulation body, or a meeting of experts in ethical issues, they approve your clinical, it's not clinical trial, it's clinical research protocol. They approve you as a principal investigator, and then you can use human tissue for research. But you cannot buy it, and you have to go through that process. So they're very strict in the use of human tissue and derivatives of human tissue. But yeah, that is more challenging than doing research with human tissue in the US. You have to go through some processing here, but there you cannot even buy human tissue. You have to do all that process. We did that, and we started culturing human cells in our lab.
That in Argentina there is a huge business cloning horses.
Yes, there is. Yeah.
I wonder if these two things are somehow connected.
Mostly in Argentina, all the agroecological field is huge, and there's a lot of research, cutting edge research in that field. I'm not very familiar with the cloning process, but yeah, we know the company, and they're very known in Argentina, and it is a very interesting project. Regarding that, there is practically no regulation when you are using another animal tissue that is not human. So that is why those companies can work in that field. I'm not very familiar with a specific regulation, but if you want to try new therapies for animals that are not human, you can do it very easily, or more easily than with humans.
Yeah, I've recently talked to a guy. They have a startup in Dubai, and they're thinking to move to US. So what they are doing, they're analyzing animal semen. They developed an app that uses AI. So they built their own model that allows you with a only microscope and your phone to analyze animal semen, starting with camel semen.
Yeah.
So, and I asked them, guys, but why not humans? And
That's right.
he told me exactly that. He told me, first of all, unexpectedly, the market for animals is much bigger than for humans, even with IVF and all this stuff. And second, that regulations are so strict. Even for human semen. I mean, it's not even a human yet.
That's right.
Yeah, that with animals, it's much more viable as a concept.
Yeah, so, sometimes even you start trying new technologies or new therapies, mostly in the orthopedics. You can start with animal applications, and that is a big market. At least in Argentina, it's a huge market. And then move to human, but when you get all the funding from selling in the animal field, you can go to a human. But sometimes even it doesn't make sense to enter to a human applications field because of all the regulations and the expensive process.
Fascinating. Okay, let's go back to the bioprinting.
Yeah.
So we already touched it a little bit. Tell me step-by-step process. How are you doing bioprinting?
All right. The main idea is to create human tissue and organs for specific patients. The process is we take a small biopsy, a small tissue sample from the patient, ideally the same tissue that we want to replicate after. So, for example, we want to replicate an ear, so we take a small tissue sample of cartilage from the specific patient that needs it. Then in the lab, from that specific small tissue sample, we isolate the cells from all the structure of the tissue and then reproduce those cells in the lab. It's called expanding. So we expand the cells to get a lot of cells from a small tissue sample. And with those cells, we combine it with any material that we can use to create the structure or the scaffold or the matrix for this tissue. Usually it's collagen because collagen is the protein that makes the structure of our tissues. So we usually use that combined with other materials. But basically we put inside a syringe the collagen with the cells from that patient, a lot of cells, and then we put this syringe in the bioprinter. And then we have to build any geometry, any specific geometry from the anatomy of that patient. So in that case, we can make a 3D scan because it's an outside structure. If not, we can use a CT scan or an MRI and then build a 3D virtual geometry of the tissue that we want to create. We send the bioprinter the geometry from the anatomy of that patient, and the bioprinter already have the syringe with the patient cells. And then the bioprinter, layer by layer, builds this geometry. After that, it's not ready for going back to a patient. We are in a very early stage. After that, you have to mature the tissue. So that is going to go to an incubator, and we are going to give this tissue all the conditions that needs to colonize the whole structure. So then it's going to be ready for the implantation, we hope.
So you are not filling all the structure, you are building a carcass for that, a scaffold, and then you let it grow by itself to create a full tissue.
Actually, the scaffold usually is solid, but it has little channels inside. So it's not only the outer part, but the inside has already cells also. We have different channels inside to get the cell culture media, like the blood, to get to the cells that are inside the structure. But it's full of cells, yeah.
You mentioned that it's nice if you can take the same cell type that you want to grow. Can you take another one, and how will you turn it into a different one if it's not a stem cell?
Yeah, for sure. You can use stem cells or you can use already differentiated cells. The cells come from stem cells and then differentiate to different cell types. For example, skin, bone, muscle, etc. You can convert a cell that is already differentiated to a pluripotent cell and then reconvert it to another cell type. Usually this is done with fat tissue because there is a lot of fat tissue that we can take without any...
What is a fat tissue?
Fat tissue.
Fat tissue, yeah.
So you can take fat tissue cells and then reconvert it to pluripotent cells. That is induced pluripotent cells, so iPSC. Then you can reconvert it to the cell type that you need. The thing is, and why we use already differentiated cells, is because we want to get to the patients very fast. And the use of stem cells or not differentiated cells has mainly two potential risks in our application. One of them is the actual clinical application that those cells can convert to anything. So the clinical application in stem cells right now is growing. So we have to see how the results are before we go to the patients with that. And the other thing is during the bioprinting process, those cells are exposed to mechanical forces when they got to go through the syringe. So that could convert the cells to different type cells. So only the mechanical forces could influence the differentiation of the cells. So we have to make sure that all the cells are the cell type that we need.
Oh my God. So you start with one cell type, it's going through a syringe, and because of mechanical pressure or whatever, they just turn into a different one.
If they are not differentiated, they can be differentiated using mechanical forces. When they are already differentiated, they usually not change unless you do this process of making them pluripotent and then reconvert them. But that is outside the bioprinter.
Oh my God. I always knew that biology is really messy, but this is amazing.
Yeah, you have to be very careful with that. Yeah.
Yeah, I mean, like you can transplant something completely different. And fat tissue, I mean, like I always thought that fat is a bad thing, but actually it's a very valuable reserve that in the future can be used to do whatever you want.
That's right. That's right.
30 year, or I mean, help you like to get a new liver or lung or whatever, and all of that because you were collecting and accumulating fat over the years.
That's right. That's right.
What a relief. In the beginning, you mentioned that bioprinting is actually not only used in medicine. You also mentioned pharma, food production. I think that also electronics. Can you tell me a little bit more about these other applications of technology?
Sure. The bioprinter, at least the bioprinters that we manufacture, have a syringe in the printing head. So you can put in the syringe anything that you want. The only requirement is when you extrude the material has to stay there. You cannot use liquid materials, for example. Mm-hmm. But if it is a gel or a paste, whatever you want, you can put it in there and start printing. So the application of that is, for example, drug printing. You can create drugs for specific patients with the specific dose that they need throughout the day. Instead of taking different pills during the day, you can put everything inside, and you can create different parts of the structure with different excipients. So different parts of the structure are going to release the drug faster than others. Or even you can create complex geometries to alter the way that these drugs are released. So there is one application. You can work with conductive inks. It's called ink, the material, but actually it's not an ink. Usually it's a gel. But you can work with conductive inks, not only for electronics, but actually for tissues that really need an electrical communication, right?
Like brain tissue.
That's right. Yeah. And heart muscle, for example, that you need the contraction to be uniform, and to make sure of that, you have to ensure very tight connection between the cells. So that is another combination between the electronics application and the tissue application with the same bioprinter.
Yeah, I'm imagining like artificial hearts that is contracting with electricity. Is it electrical, the heart contraction in general? I thought it's like because the cells are like and back. No?
Yeah, but it has electrical communication.
Oh, this is so interesting. Wow. Okay. I think that your company, LifeSci, is very special because you're actually producing these bioprinters. You're not just printing the stuff, you develop the hardware. Can you tell me the story, how you created your very first bioprinter?
Yeah. So I was working in a medical devices company, an implant factory back in Argentina. And I was working on the research and development department. And I have a boss that was very nerd like me. So we spent time looking for new technologies all the time. And then I found out about bioprinting, and I thought this is huge. The human impact that you can create and the technical challenge is great. I really love challenges. So I quit my job and created the first prototype of the bioprinter, combining different parts of office printers, conventional printers, a scanner and an old disk reader. So we have that model back in our lab as a museum.
Under the glass.
To remember where we started. And yeah, we started with that. I started working with a research lab in the university that they were working on another approach of creating human organs, that was to take a living organ, take all the cells, and then repopulate with the patient cells. It's usually known as xenotransplant. So you take an animal organ, you take all the animal cells, and you put the patient cells on it.
But what is left there if you took all the cells?
The structure, the matrix.
So there is something, yes. So not only cells, but there is.
That's right. The collagen, for example, that we use to create the tissue, the xenotransplant actually is using the actual organ. There is in the middle an application when you take the organ from an animal and you take all the cells out, and you have only the matrix of the organ, and then you put the patient cells. So in that case, they were working on obtaining the collagen, and we used that collagen for 3D printing. And from there, we started innovating in the hardware all the time. Every year, we make a new model of the bioprinter based on the feedback that we get from the users. And what we found out was the academia wasn't going as fast as I would like to. So that is why we set up our tissue engineering lab in Argentina, and we started working on the actual application of the technology. We kept selling the bioprinters, but we started doing research in-house in two main applications. One of them is cartilage, and the other is human epidermis for testing cosmetics. Instead of using animal models, you can test it in human epidermis and get better results than in the animal model. So that is one of the short-term applications of bioprinting, creating the human tissue for testing things without going to a patient and testing things in the people.
So who owned the cells that you were using before you got an actual permission to take the cells from the patient and use them? Were they yours?
You can buy research line cells.
Okay.
Those are cells that are not isolated, at least not now, isolated from human tissue. It's been done a lot years ago, so you can buy those cells. But at first, it were my tissue. So yeah, once I had to deal. I'm not sure if this is gonna be published. I did a biopsy to myself.
Oh my God.
Because the people that were working at the lab, they were impressed by blood, so I had to do it myself. But we started, the first cells that we got were from me.
Scientists can go to great lengths to make their discoveries.
That's right. We are fully committed.
Absolutely. Why did you choose this name, LifeSci?
That is something that we are gonna say in a few years further. There is a lot of history behind that name. Life SI. SI comes from Soluciones Integrales, integral solutions. So, but the actual name Life has a lot of history, and someday we are gonna publish it.
There is a story. This is a cliffhanger, ladies and gentlemen. We will come back in a few years to tell you the real story.
Yeah, that's right.
Yeah, behind the LifeSci.
That's right.
Do you think that your fascination with robotics found its application in bioprinting? Did it help you on your way?
Yeah. I found out that every project that I start working on always has the combination of the technical challenge that I love. I'm very nerd in the robotics, and with the years I started being so very nerd with the lab part, the biology also. But usually those projects has the combination of technical challenge and a human impact. That is the main goal of Life SI, to make the difference through our passion. Our passion, mine and the team behind me, is around tech, different parts of the technology, biotechnology, mechatronics, etc. We all share the main goal of the company, of the project actually, is to make the difference. We are a company, we are not a foundation, but there is the goal behind all the project.
And a huge one, I would say.
Yeah, it is. It's another challenge.
I cannot go around the ear story. Please tell the ear story to our audience. So the question, when we first met with Adi gave a presentation at a conference in Vitalia, that time still Vitalia in Honduras, a pop-up city dedicated to radical life extension, to cancel death. Adi was giving a presentation, and after he finished, a lady from the audience asked Adi, she asked, what is the favorite thing that you printed? And then there was an ear story. Yeah, so our main urgency that we have is to get this very fast to the patients. So to do that in the medical field, you have to go through very solid steps. You cannot mess around. You have to be very solid in the science, get it to the patient, get results. So we could pitch, and it happens to me when I pitch to raise funds. We could pitch, for example, we are gonna build a next kidney
And that is our goal, but we have to be very real, very scientifically correct when we talk. So that is why we started with a very simple tissue that is cartilage. The idea with the ear is there are patients that are born without the ear. So usually the surgery that they have is a reconstructive surgery. They take cartilage from the rib cage, and the surgeon inside the operating room try to mimic the other side, the other ear, carving the cartilage. And then they implant it under the skin. So one of the proposals here is to scan the other one, the healthy or the normal one. You can mirror that in the computer and then use the bioprinting process to replicate that ear. And you can reduce the operating surgery time. You can reduce the quantity of surgeries because they go through 3 different surgeries to solve that. You can help a lot of patients. The incidence of that is not very huge, but there is a lot of people that really need that. And with those results, we are going to move forward to the next steps that maybe the quantity of patients is bigger. But yeah, we want to move over very solid steps. There are different strategies to solve the regulatory challenges that we have along the way. One of them is not using bioprinting, using another technique more simpler than bioprinting. That happened in the drug 3D printing process. In 2016, FDA approved the first 3D printed drug. And if you see, they didn't create any complex geometry. They didn't use a new drug. They didn't use a new excipient. So the strategy in there was, we are going to use a drug that is already approved for the application that is going to be used. We are going to use an excipient that is already approved. The geometry is going to be very similar to the commercial drugs that are made by compression. And the only thing that we are going to change is a technology in the process. Thinking about the medical or the clinical application and the regulation that is behind that, you have to go in very small steps. We cannot go to the FDA and say, all right, we're going to print with a new technology, with a new material, a full organ, and we want to implant it. Yeah, that's right. That's right. So yeah, we have to go through very solid steps and step by step. So that is going to take time, but we don't sleep too much. So it's going to be faster.
I know that at your team, you have a person who got this ear transplant, right?
Yeah, we got the first surgeon in the world that implanted a 3D printed ear. That he's here in Texas. He did it with another company. They went through, I think, the Wall Street Journal or some newspaper like that. It was a huge news. So we wanted the best person in our team. He's an advisor in the company right now. So yeah, we are very urgent to get it to the patients, and that is why we are working around the clock, and we have all the process already prepared to get there. Right now, we are focusing on the preclinical studies. You have to go through preclinical studies before you go to a patient. That is the main goal that we have.
What kind of scientific or maybe engineering breakthrough in bioprinting can you imagine that would lead to mass adoption of this technology?
That's a great question. I'm not sure if the breakthrough is gonna come from specifically from the technical part, but the thing that is gonna limit the adoption is gonna be regulation for sure. But before that, we have to solve a lot of things in the technical side or the biotech part, right? And the main one, I think, is going to be vasculate. That is why we are working on different strategies to solve that. Because if you can solve that, you are one step closer to the actual application.
So with ears, you don't need to do that, because it's
no,
like a
that's
cartilage.
Right. That's right. That is why we started with that tissue.
So what are the approaches that seem promising right now? How to connect this tissue to blood system? There
are different challenges in there because the vascularization structure, it gets to very, very small channels. So if you want to print something with very, very good resolution, the printing process is going to be very slow if you want to print something big. So you have to keep the cells alive during that process, and that is challenging. So there are techniques that allows you to print with very high resolution, but some of them are slow, some of them are fast, but you cannot use cells during the process because they use UV light, for example. So there are different challenges, different approaches to solve it. There are options to create the structure, the matrix with a channel inside, but that is not vascularization. Our arteries have different layers of different cell types inside the artery. So it's not only the tissue. So only the artery has different layers inside. So you have muscle in the artery. So it's a very challenge itself to create an actual vessel is a challenge itself.
Oh my God. But how does it happen in nature? I mean, like,
It's
if you cut
beautiful.
yourself, yeah, so how does it happen when, I don't know, like my arm grows some skin? How
Yeah.
does it happen?
Yeah, one of the approaches to promote the vascularization in bioprinted or artificially fabricated tissues are using neovascularization in inductors, I think is a translation. But there are some material, some molecules that promote for the new vessels creation, the neovascularization. Usually, these molecules are usually present on tumors, so that is why they are very vascularized. So there is a risk of using those molecules in the 3D bioprinted tissues. That is why we are not using that approach. But yeah, you can promote the vascularization with different strategies. One of them is using these induction solutions or materials that can promote the neovascularization.
Yes, that sounds like a challenge. Every time when I talk with a scientist who does biotech, I am amazed by how complex is actually human body, and this is a miracle that we even
It
can
is.
do something there.
It's crazy.
My, yeah. Do you think that we will ever get portable bioprinters? I mean, yours are not that big, but is it possible to make it even smaller?
Yes, for sure. There is one of the techniques that is called in situ bioprinting that is basically like a automated syringe or a motorized syringe where you can print inside the operating room. The idea of this technology is for patients with burn
Mhm.
lesions, for example, burn wounds. The process is gonna scan the depth of the wound, and then after that there is a syringe that is gonna be extruding the material with the cells or the material only to create like a layer and protect from infections. That is called in situ bioprinting, and it is a small device. But yeah, that is for in situ application, not for complex organs. I think for the actual complex organs, it's going to be a big equipment. We are working on that, and it's going to be bigger than the ones that we had there in Vitalia.
Wow. So what will it be? What will you print with your new printer? What are you planning?
We are hoping to create more complex organs and automate more parts of the process. Yeah, that is the idea with the round that we are raising right now is to get to the preclinicals with the cartilage and to build the MVP of this new biofabrication platform.
Can you do that faster? I mean, I've seen how it prints the tissue. Obviously, that's okay for research. Yeah, but when you would need to build, I don't know, a heart, yeah, that's quite a huge object.
Yes.
Is it possible to do that faster?
Yeah, there are different techniques in bioprinting. One of them, the most common one, is to put the syringe with the cells and the cartilage and you extrude the material. And there are others that use UV light, for example, or blue light, and you project every layer. It's like a projection of that layer. And then you go up a little bit and project again, and then go up and project again. And every point where the UV light or the blue light touches the material, then it's gonna be hardened only in there. And then you take all the material that is not in there. that process is a little bit faster. It depends mostly in the geometry that you are printing, but it is faster than the other one.
That reminds me of a visit to the dentist.
The same process. The same process.
Literally the same process. Yeah.
That's right.
Oh, that was a super interesting discussion. Tell me how people can learn more about what you're doing and stay updated about your company and your work.
Yeah. So we usually give 3D bioprinting courses for people that wants to learn about the technology, and all of our information, the bioprinting courses, the news about the field are in our website that is livesi.technology. Oh yeah, and happy to help anyone that wants to enter the bioprinting field. It's beautiful.
Absolutely. Everyone should take this amazing opportunity and come to Adi and ask him how to jump into the bioprinting.
That's right. That's right.
Thank you so much. It's been
Thank
a
you.
pleasure.
Yeah. Thank
Yeah,
you.
wishing you a great day.
Thank you. You too. Bye bye.
Thank you. Bye bye.